This is something that's actually kind of a pet peeve. Anybody know where pH is coming from?
The term pH?
Yeah. Well, okay, we'll go start with the term. What's the term pH? Huh? It is a logarithm. Okay? The P stands for the negative log, and then your H is of hydrogen ion concentration. That's why you can have pKa's, because it's the negative log of the Ka. That's why you can have POH's, because it's the negative log of the OH. Okay? When we're thinking about the pH scale, it really is based off of one molecule. What's the one molecule our pH scale is based off of? It's based off of water. And water decomposes to H plus and OH minus. So neutral water has an equal concentration of both H plus and OH minus. If it's acidic water, you have more H plus, less OH minus. Okay, so it's the balance of those two ultimately in water that allows us to determine this. Okay? So if someone asks you for the pH of an organic compound, it's really nonsensical because most organic compounds don't dissolve in water. So if you're trying to figure out the pH of an organic solution of ether and you dip your pH paper into it, what happens? Well, it comes out neutral. Why? There's no freaking water. Okay, so if you want to figure out the pH of an organic solution, you have to add water, mix it up, let them separate, and then test the water. Because whatever ions were in the organic solution will, in theory, make it into the water solution as well and get you some pH reading. Okay? So, we come into this concept of pH happening with our carboxylic acids. And the biggest reason is because when you move into biochemistry, you've got amino acids. Okay, our proteins, our carbohydrates, all those things have the acid functional group showing up in them. Okay, fatty acids. The structure of those things will change drastically if you change the pH. Okay, so what happens at low pHs? What does a low pH mean? It's a high concentration of H plus and a low concentration of OH minus, which means it is acidic. A high pH means it is basic. So we have a low concentration of H plus and a high concentration of OH minus. Okay? We'll forgo some derivations of what's happening within this in our relationships that we're about to drop into this, and we'll kind of leave you to discover that in biochemistry, or if you want to talk about it after class, I can attempt to go through and derive it so that you can see how these relationships can come about. Okay? So, if we take a look at this carboxylic acid, and we now drop it into a low pH environment, say something like a pH 1, what would we expect to happen to that structure? Okay. We would expect our structure, or our acidic hydrogen, to be on the structure. Okay. We wouldn't expect to add another hydrogen, okay, which is what you were suggesting there, Janine. Adding another hydrogen not going to very likely happen. Very low concentrations of that. What we're referencing is the acidic hydrogen on our carboxylic acid functional group. So at a low pH, we would expect that hydrogen to be attached to it. But then what happens when we move up to a pH 2? Or 3? Or 4? Or 5, 6, 7, and 8? That's why you attend lecture. When does that hydrogen all of a sudden come off of our structure?
After they match?
Okay, it has to do something with the actual acidity of that compound. Well, how do we measure the acidity of that compound? How do we quantify the acidity for any acid?
Measure the pH.
No. Once we add it to water, yeah. We would come up with some magic number, some magic relationship. And when we added that compound to water, it produces this much acid. 152. That's the end result. Because the concentration could vary depending on how much of it I put in there. If I put more of that acid compound in there, I get more H+. But that doesn't change the acidity of the compound. The compound is still that acidic, I just added more of it. So we need a way to kind of average out concentration. Comes back to equilibrium. What's the equilibrium for an acid? Okay, what term do we use to represent the equilibrium for our acids? I know, I'm making your heads really hurt.
H plus and
It involves H+, it involves A-, and it involves HA, yes. So what we're looking at is if I take some unknown acid and I add it to water, I can get H+ and A-. I want to be able to quantify how much H+ is made regardless of what A is. So if I change A, how can I quantify how much H+ comes out of this? Okay, without bringing in this concentration issue, because if we bring in concentration, I can increase the concentration of HA and I'd get more H+. So I could take a really weak acid and add a lot of it to solution and have a pH that's about the same as if I took a really strong acid and added a very small amount. It is the equilibrium constant, our Ka, 152, okay, which we could also take all the way back to the pKa. Why do we use the pKa instead of the Ka? Why do we use the pH instead of the H+? pH of 7 has what H+ concentration? Close. 10 to the negative 7th. Okay, a pH of 7 has 10 to the negative 7th H+ ions. Okay, well, which number do you like or would rather work with? 7 or 10 to the negative 7? 7. We shift everything to the log scale because it's a little bit easier for our minds to wrap our heads around. Okay? So when we look at pKa's, we can then evaluate something about their acid strength by looking at the pKa. The smaller the pKa, the more acidic it becomes. Okay, so a standard strong acid, like HCl, anybody know its pKa? It's about -7. Really, really, really small. So small that it becomes negative. What's the pKa for a carboxylic acid? It's about 4, between 4 and 5 usually. Okay, so we now have some way to quantify that acid characteristic. Where this becomes helpful is we can now compare that back to the pH. If I say my carboxylic acid has a pKa of, let's go ahead and say just 4.5. If I'm at a pH of 1, what should I do with that hydrogen? Leave it on or take it off? pH of 1. It stays on because my pKa is 4.5. So I'm in a more acidic environment than the acid that I'm working with. pH of 2. Leave it on. pH of 3. pH of 4. pH of 5. Now it comes off. Okay? So what's happened is as we change the pH, our environment can now affect our structure. Okay, why is this becoming critical importance for biochemistry? When we run a biochemical reaction, we don't just mix two things together and just run it at whatever pH it is. Your body, or all biology, sets a particular pH and says all chemistry must happen under these exact conditions and no other. Okay? Because what happens when we change outside of those conditions? All of the enzymes, all of the proteins, all of the DNA that was designed and built around that individual pH system changes its structure. Well, if the structure changes, everything goes out the door. I imagine you're walking down the street and your pH all of a sudden, and your body changes structure. Your feet are now on top of your head. Okay, that's going to be a really odd thing to deal with. Okay, so what's happening within your body as well with all these individual molecules? We're running at that standard pH, and we have to control it at that pH, which means we see drastic structural changes or structural influences when we look at these compounds. Okay? So for an individual acid, it was relatively straightforward. We just compared it straight through. But what happens if we mix it up and we now take it to an actual amino acid? What happens now? If I move to a pH of 1, okay? pH of 1, I'm at a strong acid solution. What does that mean for my structure? What do you mean remain the same? You're not wrong. The hydrogen on the carboxylic acid should stay on there. Okay? Carboxylic acids, we've been talking about them. It just belongs there. It's where it should be. But is there another functional group in this structure? What's that other functional group? I have an amine. What chemistry is available to amines? We have some acid-base characteristics. Are they acidic? No. But they are very good bases. So if I'm now in a strongly acidic environment, what should happen to that base? I should have a hydrogen attached to it. Okay. If we start increasing the pH, say let's jump to a pH of 6. Actually, we can go to pH of 7. Might as well. Neutral pH. What should happen to the carboxylic acid? Well, what was the pKa of the carboxylic acid? It's at about 4.5. If I'm at a pH of 7, I'm saying I'm in an environment that is more basic than my functional group. Which means what should I do to the hydrogen? I need to get rid of it. So at a pH of 7, that species is negatively charged. Right? Do I have another acidic functional group? The amine has an extra hydrogen on it. Okay, so to answer questions about the amine, what do we also need to know now? We need to know the pKa of the amine functional group. Okay, how many of you memorized that one? Most people haven't, that's fine. Somewhere about 9. So at a pH of 7, what should happen to the amine hydrogen? It should stay there. We can't add another hydrogen. Why can we not add another hydrogen? There's no electrons to do it, so it stays on there. What happens if we increase the pH once more? We move up to, say, a pH of 10. What happens to our carboxylic acid? Should the hydrogen be on there? No. Okay, so we keep the hydrogen off. We've got our lone pair. We've got our negative charge. How about our nitrogen? It should donate the hydrogen. I should return to the neutral nitrogen state. Okay? So what's happening at each of these stages, and this can happen in biology too, is within enzymes, you can locally change the pH. So even in the body, there are parts of our body that are at pHs in the 2 or 3 range. There's parts of our body that are up in the pH much higher than that. The one that most people remember on the low end would be acidic. Where is there something acidic in your body? In your stomach. Why would we want it to be acidic in there? We're trying to break apart all those other molecules, okay? The food that we're trying to intake. So we have to be able to generate that. Which means if we look at the cells around that area, those cells are very resistant to acid because they need to function at that acid level. Okay? So the enzymes present in our stomach are going to be entirely different than our enzymes found anywhere else in our body because they're at drastically different pHs. So if we just taken the enzyme out and just changed the pH, well, all the structure is now going to change. Okay? Because if we look at an acidic pH, what was the charge on our molecule?
Positive.
Okay, our pH at acidic conditions, we have that extra hydrogen on our nitrogen. Our overall charge is positive. If we shift to our extremely basic conditions, our overall charge was negative. Would we expect different interactions between a negative molecule and a positive molecule? Yeah, you can get drastically different interactions. Okay, so changes in our pH are of critical importance for biochem. So it's one of the reasons why it does kind of float into organic chemistry, particularly when we start talking about carboxylic acids and the amine functional group. Okay? Questions about that? What happens if you have more than just two acidic functionalities? If I add another functional group on this structure. What happens now?
That's more acidic than the other carboxylic?
We now have yet another acidic functional group. We have to evaluate its acidity relative to all of the other ones as well. Right? And so when we start changing the pH, we're going to have to treat each of those functional groups as independent structures. Right? So based on what we know already, we said the pKa of one end of this was probably wrong, but we'll just go ahead and say it's 4.5. The pKa of our nitrogen was about 9. What's the pKa of that new carboxylic acid? It's 4.5. It's going to be exactly the same. No difference in comparison to the other one. Oh, crap, it is no difference. You're right, you're right. Yeah, I know. Try again. Would we expect the exact same pKa?
Yes.
If it's yes, it has to be exactly the same. Are those carboxylic acids exactly the same? We start our hydrogen for both of those. What are those hydrogens attached to? Oxygen. What are those oxygens attached to? Carbonyl carbon. What are those carbonyl carbons attached to? Carbon versus carbon. Is there a difference between those two carbons? Yes. So is there a difference between our acids? Yes. It may not be obvious to see that difference yet, because carbon versus carbon, there really isn't a big difference in electronegativity. So let's move one atom further away. For this one, I run into nitrogen. For the top acid, I run into carbon. So my bottom acid is closer to the nitrogen than the top acid. What does that mean? What is that nitrogen going to do to this structure as far as acidity? Why does it make it more acidic? Why does it pull the electron density away? So for it to donate electrons, so that's an interesting theory. Let's look at the opposite of what Isaac was saying. You're still right. We share the electrons in. How many bonds does this carbon now have? It would have 5 bonds, so what do we have to do? We have to lose one. The electrons go right back out. So when we look at this electron donating versus electron withdrawing effect, our electron donating effect works great with nitrogen when it can actually donate the electrons in via resonance. Can we do resonance? No. So we've lost the resonance argument. What argument do we now have to shift to? Inductive effect. And induction has to do with electronegativity. Which is more electronegative, the nitrogen or the carbon?
Nitrogen.
Nitrogen. So the nitrogen draws more electron density away than the carbon, which means more of the electrons in that bottom acid are drawn into the structure. If they're drawn into the structure, what's holding the hydrogen on? It's a weaker bond, which means the bottom acid is the stronger acid. What's the pKa of the top acid? Any number bigger than 4 or 4.5. It's probably not much larger, so we could probably approximate, let's say, a pKa of 5. Okay? So we would expect to see that bottom hydrogen get removed first when we slowly change the pH, then the top hydrogen. Okay? And if you think this was complicated, biochemistry takes it to a whole nother level because they bring in this concept of the isoelectric point, which is referenced in your textbook. You are more than welcome to look at it. It's not going to be relevant for the ACS test. All they're saying with the isoelectric point is the point at which your structure is an overall neutral charge. Okay? So one quick example, the structure up here, what charge is it? Our structure that's drawn. What is the charge on that? It is neutral. So is this the point at which we're talking about for the isoelectric point? It's a harder question. What functional group is this? It's going to act as? What functional group is this? An amine, which acts as? Is the structure as drawn a valid representation of this molecule at any pH? No. Part of it is an acid. The other part of it is a base. What's going to happen? An internal acid-base transfer. Okay, so we may look at the structure and say, oh yeah, we're happy with it. And that's what organic looks at. Hey, I've got this structure as my answer. I'm happy. I can move on. Okay. Biochem looks at that and says, no. That structure cannot possibly exist at any stage in the game. Okay. What would end up happening is an electron or a hydrogen transfer. We would have a negatively charged oxygen over here. And we'd have our positively charged nitrogen over here. Why did I deprotonate the bottom carboxylic acid and not the top? Coming back to it. Doesn't change the question. Okay, who said it was more acidic? Don't blame me. You said it was more acidic due to an inductive effect. Okay? Okay, or we can blame Isaac. Isaac said it was more acidic. I just happen to agree with Isaac. Okay? So the structure that we see is this guy. Okay? This structure makes more sense because our acidic functional group has lost its hydrogen. Our basic functional group has picked up its hydrogen. Yes, I still have another acid there, but are either of the two remaining functional groups basic enough to remove that hydrogen? No. Okay? This is, when we're trying to determine our isoelectric point, we'd want to know our pH when this structure existed in solution. Okay? And there are a variety of ways to calculate that, but I don't really want to get into it. What I will address is a special characteristic of this. This is a neutral structure. When we look at it, give me a sec. If it's a neutral structure, why are those charged? They have to be charged. So it's overall neutral, even though there's charges present. A structure that has that is known as a Zwitter ion. Anybody speak German? No one speaks German? I think Zwitter is actually German for twins. I'm not positive on that, but I think that's actually true. Now, what was your question? Did I lose another comment?
I was going to say, isn't that a Zwitter?
Yes, it is a Zwitter ion.
It's in the thing.
It's in the thing there, yeah. Okay. Questions? Yes, then yes. Okay. He's seated. Go ahead.
So for those regions, the pH has to be given. Otherwise, right?
The pH is given for those questions. pH of 7. Okay, what you would have to do is look up the pKa's for each of those compounds.
Right.
When you move into biochem, guess what you have to do with those pKa's?
Memorize.
You have to have them memorized.
But for ACS?
ACS, you don't have to worry about it.
Or memorize.
Yep. Okay. So when it comes to the ACS, I still highly recommend you have pKa's of your common functional groups memorized, because that is helpful. It is very useful to have that information nailed down. If you decide to not do it, you don't need to do it. Okay? I've only got them memorized roughly within the last couple years, okay? Just from working with them often enough where I've had to check and look them up. Okay? But you do not need them to do well on the ACS test. You do not need them.
So, let me see if I understand this. The isoelectric point is like a Twitter ion at a certain pH.
Let's see. When it comes to biochem, yes.
At the given pH.
Okay, it is, the isoelectric point is the pH at which your structure is overall neutral, is the definition. Okay, so in this case, the point at which our structure was neutral was the Zwitter ion. It does not always have to be like that. Okay? And it doesn't always have to be pH dependent. There are other structures that are Zwitter ions. For instance, this structure is a Zwitter ion. What is that compound? It's nitrobenzene. I don't see any ions in that. How do we know it's a Zwitter ion? As you draw out the Lewis structure, and our Lewis structure of our nitro functionality is it's a Zwitter ion. Okay, overall neutral, but still charged. Okay? I don't know that that term would show up. In fact, I'd be kind of surprised just because we're Americans and we don't like Germans, apparently. Okay? I think I even reported that. That's even better. So, our lesson out of this, which we already kind of alluded to, if your pKa is lower than the pH, yes, then we need to remove the hydrogen. Should not be on your structure. If your pKa is higher than the pH, then keep the hydrogen on there. Okay. You realize that that is very easy to confuse because we could swap pKa and pH, and we'd have to change our higher lower statements as well. Okay. So you can think about it as acidic or basic. That might help as well. Okay, so I try to just include those rules up there just in case. When it comes to the synthesis of acids, we've seen a bunch of syntheses already. Okay? These are old ones that you've already seen. We could add chromic acid to a primary alcohol. When we do that, we end up with the carboxylic acid. See, did I stepwise this? Whoa, I didn't stepwise it at all. Awesome. So behind that little box is a carboxylic acid. Bless you. And we would end up with our acid. Permanganate, okay, or chromic acid with a benzylic hydrogen. So as long as there's a benzylic hydrogen, we'll end up with benzoic acid. Okay. If you're looking at the handout that I gave you on Tuesday, that's good, because this is just a copy from that. Okay. I did it, though. I still drew them. I didn't just copy them somewhere else. Ozone goes through and cleaves open your triple bond, just like it did with an alkene, but with an alkene, we have the option of keeping a hydrogen or not. With ozone, or with an alkyne, there is no hydrogen to keep or remove, which means you have to replace that bond with an oxygen. And we end up with the diacid structure, where we've completely ripped the two structures apart. I don't remember how many carbons I had. And we've got our two acids. Okay? So these are reactions that you've already seen to make these. Does anybody have questions about them? Yes.
Number 2.
Yep.
So we lost 2,
You lose the 2 R
the
groups.
2 R group.
Yep. As long as you have a benzylic hydrogen in that second step, as long as that hydrogen is right there, you will immediately turn that into benzoic acid.
So it's regardless what we have the R group here right
It does
now.
not matter what the R group is. Completely irrelevant.
Where do they go?
Into solution. Okay, you're not responsible for knowing what happens to them. What if we drew out this structure? What happens now? Why? I see hydrogens there. Why don't those hydrogens turn this into benzoic acid? Those are not the benzylic position. You have to have the hydrogens at the benzylic position. Okay? I don't have any actual things written here. I just kind of had to make this one up. So how else can we make acids? Can we make an acid from a nitrile? What does a nitrile look like? Okay, it's equivalent to our cyanide. Take CH3, carbon, triple bond, nitrogen. Can I convert that into an acid? Those of you that have been showing up to the Friday study session should absolutely be saying, screaming, yes, 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 you absolutely can do that. Okay, why? If you've got those pi bonds with a nitrogen, those are extremely susceptible to an oxygen nucleophilic attack on our carbon. So all we have to do is add H2O. Okay, maybe an acid catalyst to help initiate this thing. The water attacks, and it'll go through a sequence of reactions, all with a bunch of protonations and deprotonations and all sorts of other exciting stuff, and we'll end up with our carboxylic acid and ammonium. Okay. Why do we end up with ammonium and not NH3? It's a tricky question. We're in an acidic solution, which means the basic NH3 would need to pick up a hydrogen and become ammonium. Why did we end up with a carboxylic acid and not the negatively charged oxygen? We're in acidic solutions. Okay? So watch out for that when you look at those overall reactions. As soon as you see that nitrogen show up in a reaction, if you're under acidic conditions, you better protonate the heck out of that nitrogen. Okay? If you're under basic conditions, what can you do? Okay? If it was O- conditions, if we'd used instead of H+, we ran under basic conditions, OH-, what would our product be? Negative charge on the oxygen. And what happens to our nitrogen? Our NH3. We won't form NH2. Okay? NH2 minus is a wicked strong base. OH minus is weak in comparison to NH2. Okay? So in the presence of just a weak base or a strong base catalyst of OH minus, we'd end up with the NH3. Okay? Can we make carboxylic acids from Grignards? Of course, that's why I asked. Okay, so if we start with a Grignard reagent, CH3MgBr, how can I turn that into an acid? As a hint, there's 2 steps. First thought, what do Grignards act as? Nucleophiles and bases. If we're going to make a carboxylic acid in the presence of a base, what's going to happen? It's going to react with it and be deprotonated. If I want to make the actual acid, what do you think has to happen in that last step? We'll have to neutralize. I'll have to put a proton on it. So the reagent in that last step is an acid, H3O+. Any ideas what you add in that first step? Got to be a carbonyl. We need that carbon double bond oxygen. You want a hint?
An aldehyde?
It's
Ester?
nope,
Or ketone?
nope. Okay, if we added an aldehyde, there's that hydrogen. Is there a hydrogen bound to our carbon in our carboxylic acid? Nope, aldehyde doesn't work. Try an ester. Not a bad idea. Then we have that OCH3, the reasonable leaving group. But if it's an ester, we'd have another CH3 coming out the other side. If we did just a straight ketone, then we have CH3s coming out both sides. Formic acid is going to give you an OH, and the issue with formic acid, nope. What was that? We have an acidic hydrogen. That acidic hydrogen is going to quench the Grignard reagent because the Grignard will act as a base. So we can't add in an acid. Maybe those of you were talking about an ester, we're talking about the ester from lab. Okay. Still doesn't work. Your Grignard's just going to attack that tons of times. You're going to get tons of carbons on there. What was that? CO2. What happens now when the Grignard attacks? Pop one of those pi bonds open, and we end up with CH3 bound to our carbon, single bond to our oxygen, double bond to our oxygen. There it is. Okay? So you can react Grignards with CO2 to make acids as well. Okay? Questions on that? Okay, that is pretty much all of what Chapter 19 is about. You'll probably see some more kind of acid-based biochemistry mumbo-jumbo in that chapter. You're more than welcome to read through it. It will probably be a decent prep for biochem. Okay? But beyond that, you're not going to see it on the ACS, so I won't ever ask you any questions about it either. Okay? So, acid derivatives, which is Chapter 22, I think we decided. Kind of a long chapter, but it really is just a repetition of what happens with your carbonyl chapter. Okay? And that, what do we have in all of these functional groups? A carbonyl. Okay? So again, repetition of our carbonyl chapter. And what's of particular importance for all of these carbonyls? They all have electrophilic carbons. So that is the primary chemistry addressed in this chapter. Electrophilic carbon, throw a nucleophile at it. Well, guess what? A new functional group, throw a nucleophile at it. Oh, you want to make another one? Throw a nucleophile at it. Okay, all it is just throwing nucleophiles at these. That's all we're doing for each and every single one of these. Nothing particularly interesting beyond that. Okay? Part of this kind of chapter is recognizing the names of these. That first one is referred to as an acyl halide, or more accurately, in this case, it would be an acyl chloride. Okay? You might see it referred to as an acid chloride as well. Why acid? Because we kind of have a pseudo weird looking acid position there. In general, what you'll find out is when we do our acyl halide formations, that we generate strong acids in the process of making them. Okay? So you'll also see the acid functionality show up. Yeah?
Are these common names, or do we also use these in IUPAC names?
It's a mix of both. Okay? So for that first one, it's just an acyl. And that ace is coming from acetate. There's our common name. okay, in purple. That whole section over there is an approximation to our acetate. Okay? If you see it in a larger structure that's not just an acetate, you'll usually see an OYL and then the halide. So, for instance, pretty sure I got the name right on this one. Benzoyl chloride. Okay. So if you're seeing chloride or any halide name show up at all, and then something else that you would say is a complex looking structure, you're looking at an acid chloride or an acyl chloride. Okay. So more than likely you won't be expected to know the name for any of these. I'm just trying to give you a primer on what's happening with some of that nomenclature. That next functional group is an anhydride. You'll sometimes also see it referenced as an acid anhydride because what whoa, don't do that. Come back here. Because we have darn close to an acid there, and we almost have an acid coming out the other side, except we're missing an oxygen and 2 hydrogens. Anhydride, no water. Okay? So that's what we've got there, is an acid anhydride. The functional group is named primarily for that middle section. This piece right in here. So if I wanted to get a little bit more fancy with this name, I could refer to this one as dimethyl anhydride. Why dimethyl? What are the pieces on the outside? Methyl groups. Okay. Next one down. What's the functional group on that one? You all know that one. It's an ester. We already talked about the nomenclature on that. You'll name the piece that looks like your acid or closest to your acid as your O8 ending. And then the R group is a substituent on it. So let's just leave it as that. Substance. Okay, and we'll get our YL in there. The next functional group, you may or may not remember. It's an amide, super relevant for organic chemistry, or sorry, for biochemistry because that is the bond connecting individual amino acids to each other in proteins. Okay, it's an amide bond. I've got some examples for naming that on the next slide, I believe. And then that last one, yeah, I was trying to come up with some fancier way to name it, but I don't really have one. It's the conjugate base of our carboxylic acid. Okay? When it comes to naming those, you might see them named with salts. So the example we looked at on Monday was potassium, in this case acetate, or ethanoate. Okay, so you're naming it kind of like an ester with something other than a hydrogen attached. So you should name whatever that piece extra is. Okay? So most of this chapter is focused on these functional groups and how we can interconvert between them. Okay, before we even get in, let's just look at the nomenclature. I was positive I'd stepwise this too. Okay, well, there are all the names. So, what are the names for these things? It was brought to my attention on Monday, something about N, and we talked about the N in class. Okay, I don't know if you guys remember, I had N-butyl written in class, and what did that mean? It's normal, so we just had 4 carbons attached to something. So our N meant normal, just to give us a straight chain carbon. Okay. The N that was actually being asked about was not a lowercase n, it was a capital N. Okay. You'll notice that capital N shows up in these top 2 examples. What is the capital N referencing? It's referencing where our substituent is located. We have to have some way of labeling where those methyl groups are located. And since the nitrogen is not part of the carbon chain, I can't number it like we would normally. So that's where you bring in the N. So in that first case, it's just N-methyl, okay? Because there's a methyl group attached to the nitrogen. What if there were 2 carbons? Sorry, let me clarify that. Two carbons. N-ethyl. Okay, so we just name whatever that piece is as an extra substituent. Yes? If we have lots of different nitrogens in our structure, that drastically complicates how we would go through and name it. Okay? You won't have to worry about that. But any ideas how we might label that? Okay, if there were multiple nitrogens in our structure with multiple carbon substituents on them. But you have to specify where each and every single substituent is, so it's very hard to just drop it in order.
You have like go down the chain like N1, N2.
Yes, it's effectively what it would become. So what you would see in your name, for instance, if we had 2 up in this case, instead of it being just NN as it is, if we had 2 nitrogens with methyl groups, you'd have N and N prime. Okay, which is obnoxious to deal with, but it does pop up every so often. In biochemistry. It really doesn't pop up all that often. You won't have to worry about it. Why do you typically not have to worry about nomenclature in biochemistry? It's not even, it's a little bit worse than that, actually. It's not that biochemists don't care. They have no naming system. None. Which means, how do you know what the enzyme is? Whatever somebody decided to call it, that's what they called it. Okay? So if you have succinate dehydrogenase, you have to know what dehydrogenase means, and it's in reference to the molecule of succinate. If you had malate dehydrogenase, well, it's a dehydrogenase again, but it now applies to malate instead of succinate. Okay. What's that? You have a morphine dehydrogenase. It's ultimately doing the same thing. So you get a lot of different enzymes doing the same type of chemistry, like a dehydrogenase, but it can only work for one type of molecule, which means you get 50 billion dehydrogenases because they can only apply to one molecule, maybe 2 or 3. Okay. What makes it even more confusing is dehydrogenase makes it sound like, what does your enzyme do? Dehydrate, as in remove water. Sounds reasonable. That doesn't necessarily hold true. It's how it was discovered, and when they discovered it, when they saw it acting, it was removing water. But your enzyme not only catalyzes the forward reaction, it catalyzes the reverse. So a dehydrogenase may very well actually hydrogenate something. Okay? So yeah, we'll leave that for your biochemistry class. Really obnoxious. Last example with if we had 2 substituents on a nitrogen, we'd have to reference the location of both methyl groups. So just like if we had a name with 2,2-dimethyl, we have to specify the location of every single methyl. There are 2 methyls. They are both located on nitrogen. We have to call it NN-dimethyl because it's specifying the location directly for each methyl group. Okay? The last one that you might see show up as a fairly common name, if we take a look at acetic acid, okay, we've got our structure of acetic acid. The anhydride is we take 2 molecules of that acid and we smash them together. Okay? That gives us the anhydride functional group. But what acid did it come from? Acetic acid. So what we'll do is maintain the acetic part of the name and just call it an anhydride. So if we had propionic acid, now I wanted to turn it into an anhydride, what would the anhydride name be? Propionic anhydride. Okay, or propionate anhydride. Okay? So it's just a question of kind of smashing those names together. You're kind of responsible for those functional groups. I don't think you'll be responsible for naming these. What you're responsible for is knowing how they react. Which gets us to our reactions of acid derivatives. Those of you in lab, all of you recognize this, right? Because you all read the pre-lab nice and thoroughly. for that ester lab that we just did. Okay. What we've got within this figure is a ranking of all of our individual acid derivative functional groups. We've got our acyl chlorides, our anhydrides, our esters, our acids for that matter, our amides, and then our conjugate base. Okay. As we'd already kind of alluded to, they're ordered according to electrophilicity. The acid chloride, way up at the very top, is our most electrophilic. Why would it be the most electrophilic? You've got an excellent leaving group in the chloride. Both the chlorine and the oxygen are electronegative, drawing electron density away from that carbon. That structure is absolutely the most electrophilic. When we move to the anhydride, we've now brought in oxygen, so not quite as good of a leaving group as our chloride. Okay, why is it more reactive than the ester? Okay, not because it has a carbonyl.
It's 2 oxygens.
Your ester has 2 oxygens.
But the other oxygen,
What was yours? What was your try again?
The 2 carbonyls.
Kind of has to do with the 2 carbonyls. One of the reasons why we said our acyl chloride was so good was why? We have a good leaving group. If we compare the leaving group of the anhydride to our ester, which is the better leaving group?
the anhydride,
The anhydride is a better leaving group because
resonance.
there is resonance to stabilize that leaving group ability. Okay, so the anhydride is more reactive than our esters because we have resonance stabilization of our leaving group. Okay? Sorry, it was balance. And this kind of goes downwards all the way through. Why is the amide even lower than the ester? Nothing? No ideas? What about resonance with nitrogen? But you're saying the oxygen can't donate those electrons?
Because nitrogen
Oh.
negatively charged, less than
Oxygen is more electronegative than the nitrogen, okay, which means it does not want to share its electrons as much. The nitrogen is willing to share its electrons, which does what to the positive charge?
negative charge.
Neutralizes it, makes it less positive, so it becomes less electrophilic. Okay?
One positive, negative.
Huh?
And R2.
It would be positive just like the oxygen would be positive. Okay? So we've got this relative order of reactivity within these. You'll notice there's all sorts of little arrows and spider diagrams coming off of this suggesting different reaction pathways. And you will notice that they are not reversible. Why might they not be reversible? Okay, for instance, if I wanted to move from, let's say, the ester up to the acid chloride, why don't I just add chloride? We'd need an acidic hydrogen to catalyze it, potentially, so we could add H+. Chlorine won't stay on because the oxygen that left is a better nucleophile than our chloride in this environment. So what ends up happening is that reaction never occurs. So how can I ever get to the acid chloride? Well, to get to the acid chloride, we have to do very particular chemistry. That chemistry involves going from the carboxylic acid and reacting that with our favorite reagent, our thionyl chloride. What did thionyl chloride do? It does have a chlorine nucleophile. thionyl chloride does a substitution reaction. All OHs are turned into chlorides. Okay? Could I have turned it into a different alkyl or a different acyl halide? Sure. With what reagent? PBr3. PBr3 will actually give us the alkyl bromide. Okay, and actually, is it just PBr3? I think it's the phosphonium bromide. I think it's POBr2, maybe. I don't remember it exactly. But we can get the alkyl bromide, or acyl bromide as well. Okay? So one of the hardest parts in this chapter is looking at those interconversions and saying, how can I transition between each of these? Okay? It is very easy to transition down. It is virtually impossible to transition up. So if you're at the anhydride and you want the acyl chloride, what do you have to do? You have to go all the way to the acid. Once you're at the acid, you can then react it with thionyl chloride to get back up to the acyl chloride. Okay? So it is very tiered and hierarchically formed, okay, or organized. So you have to be careful with those interconversions. A lot of it has to do with those electrophilic characteristics. Okay? Questions about any of that? Okay, so our nucleophilic addition conversions. And each of the mechanisms for all of these reactions, when we're looking at how any of these acid derivatives react, they're all following the same basic two steps. Right? When we're looking at the acyl chlorides and the anhydrides, you pretty much just get those two steps. Right? And that's because they have fantastic leaving groups. When you get down into the esters, the acids, and the amides, we have pretty crappy leaving groups. So what do we have to do to make those leaving groups better? We could try and make the carbon more positive by doing what? How can you make the carbon more positive in those cases? We add H+, which then starts to polarize that carbonyl bond, and our carbon becomes more electrophilic, which means we have to add some kind of acid or sometimes base catalyst. What does that mean as far as the mechanism goes? Well, you remember when we looked at the mechanism for imine formation from a carbonyl and how we had 6 or 7 different steps? All of the mechanisms involving those bottom 3 are going to involve those 6 or 7 steps. Because you're putting protons on to have a nucleophilic attack, then you're taking protons off, then you're putting protons on, then you're reforming a pi bond, and you're removing a hydrogen. Okay? You've got a whole bunch of extra steps happening in there with shifting the hydrogens back and forth within your structure. Okay? So you have to be careful when you go through and do that. So what do I mean by careful? If you're running under acidic conditions, what charge should you see on your structure? Why a positive charge? Because under acidic conditions, you supply hydrogen with what charge? Positive. So if you put a positive thing onto your structure, the structure becomes positive. So under acidic conditions, we expect to see a lot of positive charge floating through our structures. What would be exceptionally bad to see? The opposite of a positive, a negative. So if you see a negative charge when you're running under acidic conditions, whatever you're doing for your mechanism is wrong, because you should not have that negative charge. Why? A negative charge is signifier for a base. Can we form a base under acidic conditions? Not a strong one. Okay? So if you're attempting those mechanisms, watch out for those charges. They can pop up on you. So, overall result is that we have our nucleophile attacking our carbonyl. Okay, so if we go through and look at the mechanism for this guy here, our nucleophile with our lone pairs on that oxygen can attack the carbonyl. Now that carbon has too many bonds, so what has to happen? We have to break a bond, but what bond do we break? If we break the chloride, how many steps was our mechanism? One step. We just said it's always 2 steps. Okay, and it's not SN2 because it's not just a direct substitution. Okay, what you will have to break is the pi bond first. Why do we break pi bonds before sigma bonds? Pi bonds are higher in energy, easier to break than our sigma bonds. The result of that first step. I'm not drawing all those carbons. Okay, we can end up with something along those lines after just a single step. Okay, this intermediate is referenced as the tetrahedral intermediate. Why would we call it a tetrahedral? Hedral. Intermediate. Why? Because we have a tetrahedral carbon in the center of that reaction. So you will always form these tetrahedral intermediates. Okay, you'll notice that even after this single step, we're also going to have to do another step. What's that other step? We're going to have to break what bond? Our oxygen to hydrogen bond needs to be broken first. So we'd end up cleaving that bond before we kicked out the chloride. Okay? So even in this relatively simple example, we have an extra step that may or may not show up in our mechanism. Okay? The official step that will always show up is we'll reform the pi bond and kick out the chloride. The net result in this reaction is that in that very first step, we went from having a pi bond to, first step, not having a pi bond. What type of reaction matches that? It's an addition reaction. And the very next step, what happens? We get a pi bond back. So this reaction is very commonly referred to as an addition elimination. Okay, so remember at the beginning of the semester I said you need to know the 5 types of reactions, and all we're going to do throughout the whole semester is just shuffle those 5 types around. Here you go. Here's a new mechanism, a new reaction. All it is an addition reaction followed by an elimination swiftly thereafter. Okay? That's why you need to know just those 5. Everything else is just adding icing to it. Okay? So what's the role of pyridine in this reaction? So we got to be a little bit careful with that. The intent on this slide is just to focus on those 2 primary steps. Okay, I think, let me check. Oh, I didn't put it up here. Since I didn't add it again, let's actually look at the overall reaction for this. I'm going to erase those. That can't happen yet. So we form our tetrahedral intermediate as our first step. If our electrons came back down, what are they going to kick out? The best leaving group. What's the best leaving group? What we just put on. Why is what we just put on the better leaving group? It's not charged when it leaves. Okay? So before we can actually reform the pi bond and kick out the chloride, what do we have to do? We have to remove the hydrogen. So we will take the electrons in the bond between the oxygen and the hydrogen, and we will give them to the hydrogen. What did we just do? We removed an acidic hydrogen. This species acted as an acid. What's going to facilitate this reaction? A base. What is pyridine? Our pyridine is our base and technically our solvent at the same time. I didn't make it more complicated. Chemistry did. I'm just the messenger. Now what happens when our electrons come back down? Chlorine leaves because chlorine is now a better leaving group because we'd end up with Cl- as opposed to O-. And now we've got our final product. All right? So this does bring up a point. When you see these arrows, you've got to be a little bit careful about what they mean. Okay? Can I, actually, I won't erase everything. Just erase the first arrows. Look just within that box. Within that box, that says how many steps? Yep. It is only one step. How do we know it's only one step? It doesn't say 2. Okay? So until you see 1 and 2, it is one step, one reaction. The mechanism may be multiple steps, but it is one reaction. Okay, so why are both things written there? Because if we look at our product, our ester, what do we care about? Well, we care about our alcohol starting material in the acyl chloride. So why write pyridine? 2 fun answers. Well, really only one fun answer. To confuse you, okay, or really to confuse people that don't go into the lab. Okay, but if we go into the lab, why do we want that second thing written there? We need to know what solvent to run the reaction in. Okay, so it's acting as a solvent. Okay, so usually what's written underneath that second arrow can act as a solvent. What else can be underneath that arrow? Catalysts. Very oftentimes you'll find catalysts underneath the reaction arrow. Right? It doesn't mean they're always there. Remember, we can shuffle these things pretty much wherever we want to within the reaction or within that arrow space. Pyridine in this case happens to act as both a solvent and a catalyst. What else might we find underneath that arrow? Reaction conditions. What temperature do we run the reaction at? Okay. So don't just assume because it's there that you have to use it. You may not have to use it. Okay? So don't panic about seeing something there. Focus on where you find the chemistry. Okay, the oxygen is for sure our biggest negative. Our carbonyl carbon is for sure our biggest positive. Put those two together, do whatever you got to do to neutralize them. There's your answer. Okay? Questions? Don't look too excited. I might think you're having fun and not tell you when class is over. Overall summary for our acyl halide reactivity. That was not what I thought it was. Okay, so what this is showing is once we've got our acyl chloride, it'd be nice if we actually highlighted it. We're now at the top of our tree. I can make anhydrides by reacting them with a carboxylate. I can make esters by reacting them with an alcohol. I can make amides by reacting them with a nitrogen-containing compound. All of those are now accessible to me. So our acyl chlorides can pretty much do whatever they want. Okay, so we get all of that kind of standard chemistry coming with it. All we've got to do is supply that nucleophile. Okay? What's that? That's any acyl halide. Okay, so that doesn't have to be chlorine. It can be X. Okay. Why do we use the acyl chloride? The only reagent we've really talked about for making our acyl chlorides is the thionyl chloride. Okay, so that's why we use the chlorine most typically. Yes?
How can we make from the acyl chloride? How can we make the anhydride?
How do you make the anhydride? Okay, so let's follow the arrow to go from the acyl chloride to the anhydride. Where's the anhydride?
Oh, you mean that? Okay.
What reagent do we need to go to the anhydride? It's right over the arrow. That's it. That's what you need to add. Look like a bunch of spears killing something. Yes?
What is thionyl chloride abbreviated as?
SOCl2. So that guy at the end there, thionyl chloride. Okay? Are there other nucleophiles? Have we ever seen other nucleophiles? What's that? No? Yes? Yeah, okay. What other nucleophile? Carbon nucleophiles. Where do we get carbon nucleophiles? Grignard reagents. What are Grignard reagents going to do when they see an acyl chloride? So we take our carbon nucleophile, boom. We'd end up with this structure, right? What's the issue with that structure? It's reactive. So our Grignard does what? Reacts a second time. So when we react with Grignards, we can end all the way down to our alcohol. You mentioned alcohols as nucleophiles. It's a good point. Alcohols are nucleophiles. That's why they're already on the chart.
I thought we were just going down the chart.
What other nucleophiles can we get? NaOH. NaOH can take us all the way to our acid. That's effectively adding water on the inside here. I don't know why that water is there. That should be crossed out. What other nucleophiles are there? All of our oxygen nucleophiles are already on the chart. Yep. Nope. In general. CN, we can have nitriles. Nitriles can react with this. What else could react? What other nucleophiles are there? Hydride nucleophiles. Are there different sources of hydride? Yeah. Could we use borohydride? Borohydride only reacts with aldehydes and ketones. It wouldn't react with this. What would we have to add to react with this? LAH, a lithium aluminum hydride, okay, which would add 2 hydrides, okay, and we'd end up with an alcohol. Or we could add dibol. What does dibol do? Only adds 1 hydride, which would make it an aldehyde. It's only a ketone if you got 2 carbons. It's a good question. I don't know how H2 would react with this. Probably not very cleanly. Okay. All of those are sort of summarized, well, sort of summarized on the slides. There it is. There's our summary on our slide. Okay. Spiders all the way out. Okay. As soon as you've got an electrophile, boom, throw in a nucleophile. So all of the nucleophiles we talked about this semester can react with our acyl chlorides. Okay. So this just kind of summarizes everything. Okay. One of the things that I would kind of draw your attention to would be this last one. What's weird about that one? What type of nucleophile are we doing in that reaction? It's a carbon nucleophile. Very similar to Grignard. But what happened when we added a Grignard reagent? It did it twice. Why in this other case does it only do it once? Less reactive because it's not a Grignard reagent, it's a Gilman reagent. What was special about our Gilman reagents? They're less nucleophilic. They will react only once with the acyl chloride. And we can get to what Lulu was asking for, a ketone. Okay? Shall I keep going? I was being kind of mean. We are done with class. Okay. The rest of these slides are pretty much the same kind of thing. Here's an acid chloride, or an acid now anhydride on the next one. How do we make, our acid anhydrides, how can we make them? Sorry, not how we can make them, because we can only make them from our acid chlorides. Then what can we do with them? Well, if we add an alcohol, we end up with an acid. If we add an amine, we end up with an amide. Okay? So again, pretty much the exact same chemistries can come out of your anhydrides. And again, the summary of all of the pieces and the nucleophiles and the stuff. All right? All right, then we've got esters. Esters are relatively unreactive. I will show one special mechanism next week. That'll be the Fischer esterification, just because that's one of those big, long, put the hydrogen on, take the hydrogen off, put the nucleophile on, take the hydrogen off, put the hydrogen on, now kick the leaving group out, take the hydrogen off, put the hydrogen on. It's one of those big, long, obnoxious ones. So I'll show that one in its full, gory detail next week. There's our summary of pretty much everything according to a different book's format on how you can interconvert between things. Yeah, it looks pretty obnoxious. I don't really like it either, but it is what it is. And then we'll finish up the amines. And I just want to say this. Anybody get it? Amines, the basics? Why is it funny? Because I'll write in the eyeball. Because amines are bases. Ha ha ha, the basics.